the study of the biomechanical processes behind extensive prey processing in salmon fishes, or kind of easier to say, I'm just going to explain to you tonight why my mother calls me a fish dentist. Um, First off, the only reason that I do anything that I do is because I think that teeth are fascinating. All I wanted to do with my life when I found out what research was look at some dope ass teeth in some cool ass fish. And if you're not aware, teeth are going to be some of the most morphologically diverse structures within the animal kingdom. That's great for us because with all these forms comes an understanding of their associated functions. And Carly, if you're watching this, yes, I know that that is a lamprey at the top left. Uh, but I mean, it's a cool mouth. I couldn't leave it out. So, with our understanding of the different functions of these teeth, there's so much more that we can understand about things like the development or the dietary ecology that is going to be associated with whatever particular organism you're looking at. That's what we write in our grants, but to be honest, I don't really care about all that. Like I said earlier, I just think that teeth are beautiful, and I want to say that their beauty is what got me interested in research, but that's not necessarily true. My first experience with research came when I met my undergraduate advisor, Dr. Phil Moda. Wow, that is loud. I know we can turn off the pinging for the people coming in. Katie, I don't know if you can hear me. Hello?
Done. Sorry about that.
Yeah, it's all good. Do we know if there's a way we can turn off the pinging for the
Oh, it's off now. No more.
Awesome. I invited a lot of my family members and other people that I love so much, and we typically run on CPT. So it's great to see everybody. If you're here, thanks for joining in. But like I was saying, so my first experience with research came at the University of South Florida in good old Tampa, just 10 minutes away from the second largest Confederate flag in the world. And I started taking the class because I was very interested in a nice young woman who suggested we take it together. And I, of course, wholeheartedly agreed because I had a invested interest in scholarly pursuits, as I often do. And I'll never forget my first day in the class. Bill had these three amazing slides up about tagging trips and the work he'd been doing with Cheryl Wilga in relation to bamboo sharks. And when I saw the class, even though I was only in there to talk to some girl, I was like, man, this is exactly what I'm going to do with the rest of my life. And thankfully, after sending a very embarrassing email that Phil still sends to me to this day, talking about how I love sharks and I basically want to know what dolphins dream, I started to get more involved. And to this day, I kind of think of Phil as not only a mentor, but as a friend that I owe a great debt to, who was amazing at karaoke and other things that would make an undergraduate really enjoy the research experience and doesn't seem like it's just full of stats and computers. Since I put this picture up of Phil, though, I will also put up some pictures of myself where on all of our trips to different various locations for our research, I was always out cold. I don't know what it was. Something about just going to conferences that makes me tired. That I digress. So I was working with Phil, trying to get my feet wet, so to speak, and let me in the lab and suggested I apply for other research experiences through courses like the Keys Marine Lab, where we could go and practice research methods for collecting and tagging different areas. We learned how to do dive techniques such as 4x4 squares and other things that I thought I'd be using for the rest of my life as a researcher, but to my dismay, that was actually the last time I dived. But what Phil was really good at was finding what students were interested in and using those interests to get them to where they wanted to be. From this picture alone, you're going to see that there's 4 PhD students, 3 vets, and a couple of other wildlife jobs, because when we came to Phil with what we wanted to do, he accepted students that he knew we had passion, and then he built that passion and got us to take what we knew, what we were vested in and interested in, that we wanted to see in the world and understand about nature, and turn that into something we could do with the rest of our lives. So because of that, I applied to the University of Washington's Friday Harbor Laboratories, REU, as Katie mentioned, thanks to Phil encouraging me to pursue my research interest. Unfortunately, this was during the case of the Trayvon Martin trial. And I remember distinctly when the verdict came out in 2013 that it was a few days before my animal physiology exam with Dr. Steve DeBan. And I unfortunately did fail the exam. So I applied for the REU program, and thankfully I was accepted, and I was able to work with Dr. Crofts and Dr. Adam Summers, as well as Dr. Victor Iyengar, all pictured here doing what they do best, just being amazing people. And I will say I never felt more comfortable and represented in science than I did in this program here. It's also where I met Matthew Coleman, who is now teaching the fish class as he does best, and found out that we look exactly alike. I also found a number of good pictures to use on my Tinder profiles. But like I was saying, so my time at Friday Harbor was spent testing the effects that tooth arrangement had on crushing excess by employing a model-based techniques. So we created these bio-inspired tooth pads to test crushing performance using a materials testing system, and all of the different arrangements we got for these teeth came directly from nature. Following that summer, that was also the year that unfortunately Eric Garner was murdered by police. No criminal charges were put forth in that case. John Crawford III shot in Walmart while shopping for things for his newborn child, as well as a BB gun on the aisle, and he was murdered by a police officer in plain clothes. No charges were filed. Also the summer that Michael Brown was shot with his hands up, some say sparking the Black Lives Matter movement along with Eric Gardner. And the same year that Samir Rice was killed in Cleveland by a Cleveland police officer who had previously left another department because of disciplinary actions. Anyway, where was I? Not anyway, but where was I? So I tested the effects that tooth arrangement had on crushing success using these bio-inspired tooth pads. What we found was actually very interesting. When placed in a linear arrangement similar to our molars and the chelicerae of, say, a crab claw, where you're having these different kind of bumps or malariform bunodont shapes, there's going to be a normalizing effect to the tooth morphology where no matter how concave or convex your tooth was, how many cusps, the radius of curvature for more malariform teeth, the overall effect of their crushing success was flat throughout. And while we're still working to explain why that might be for the REUs right now, I did my REU back in 2014. I'm still working on that paper to this day, so don't feel bad. You know, just kind of keep going at it. You don't fail until you stop. Stephanie, I promise we're gonna get that thing published soon. Don't let good results die. I took those positive results where we found something interesting and I used it to fuel my research back at the University of South Florida, where I began my own research project that I started from start to finish. I had to find a fish that I knew I could get my hands on easily since I had no funding. And so I turned, of course, to an invasive species. The fish that I picked, Pterygophilus disjunctivus, is an invasive species all over Florida, and it was so easy to find them. I could just go to different lakes and ponds on campus, reach my hand in and grab a couple of them out. I spend a whole bunch of days in between classes casting with the fish net in these pristine looking ponds because all of these catfish were just riddling the banks of the water, and it was perfect pickings to have. I don't know, I think I had at one point like 6 or 7 of these things in the lab. And what I studied was their ability to simultaneously feed on algae that covers rocks while still adhering to those surfaces using an oral disc pad that was inside of their buccal cavity or their mouths. And since my work only focused on the subambient pressures that were creating their mouths, more so than the teeth, so I'm not going to get too much into this today. But I did think it was a really good chance for me to take what I'd learned from Friday Harbor and take the passion that comes with being at a research lab and being in such an intensive course and apply it to the rest of the school year where I could then coast off of that passion into a project that I was very happy about that I got a chance to present at my own university. While working on these fish, I remember that I had no luck getting them to feed. And so I would try to feed in the evenings because, you know, when you can't get the fish to feed in the day, somebody tells you, oh, maybe they're more active at night. And so I would sit in lab under like this red light camera for the longest, trying to wait for the fish to do something because they're supposed to be more active. And I was driving home at around like 2 a.m. I got stopped for a dim headlight and had the following exchange with an officer. Thanks to all the research experience I had, I feel like it prepared me for my current position as a graduate student under Dr. Elizabeth Brainerd. And if you know Beth, and I think she's probably in that audience there right now at Friday Harbor, you'll know that Beth is kind of the GOAT. And she does not play around when it comes to research because she picks people on her team that she knows are passionate. As soon as I graduated from undergrad, I was almost immediately thrown into the fish class myself, like I told the fish class earlier today when I had a chance to talk to them. And so that summer after I graduated, I think I spent a couple of weeks or maybe a month kind of doing my own thing, sowing my oats and going to Greece or something like that. I don't remember. And then I immediately came to the fish class where my project was looking at the feeding apparatus of striped surf perch to understand the function of a throat muscle known as the sternohyoides. So fish do this thing called suction feeding where they create a negative subambient pressure inside of their mouth so that they can draw in both water and the prey that they want to capture into their mouths using a combination of their axial or body muscles as well as their cranial muscles. Now the sternohyoides has a bit of dual functionality where in some fishes it is one thing and other fish does the other. And so we were looking to see exactly what it was that it did in this particular fish species. I'm happy to say that as of like 3 days ago, that entire paper was accepted. I'm not sure how much of my figures I can put in the talk because it is now at the stage where it's fully accepted and I just need to go through, finish up all the words and get the proof ready to go with the editor. Like I said, that was my first month in grad school that I worked in this project, or the summer before my first month, I should say. And one of my first experiences at my university was with a professor who I think he thought he was being nice or ultra woke. It was something new that I'd never experienced because living down south most of my life, the extreme kind of consciousness and the understanding of social and racial justice theory that is very prevalent in northern states, or actually more kind of liberal-leaning and left-leaning towns, was something I wasn't really used to. And so I didn't know what to do in this particular case where a professor never met me before, did not know my name, decided to trap me in the hallway and talk to me for a solid 15 minutes about race theory and how I was likely whistling because I wanted to disarm people given my size and stature. This was also the semester that President-elect Trump was elected, as the name suggests. Startling. I don't really got to say anything else about that. It also led into my second semester of graduate school, where instances like this became the norm, and it was more so getting used to just kind of keeping my head down in places and spaces where I wasn't necessarily what people were used to seeing. And we're back. So the fish with human teeth is basically my dissertation, where I'm looking at this monstrosity of the fish, the red Belly Paku, a member of the Serrasamid family, which is the same family that contains your kind of notorious piranhas. And they consume, the Pakus consume a primarily herbivorous or granivorous diet with these weird incisoriform like teeth. My whole dissertation started with the question, if teeth can give us an extensive understanding of the organism that possesses them, just what do these teeth say? In fact, when I found the particular teeth in question for this species, they were isolated as they were in this picture here, and I thought that they had to be mislabeled because I was like, there's no fish with these weird kind of mammalian teeth in their mouth. This just doesn't exist. Of course, as my life typically says, I was wrong, and so it started an entire program where I've been looking for a while at this strange family of fishes. And the deeper I dug into the clay, the more interesting they got, as a degree of occlusal offset that you're seeing in this fish family varied from extreme overbite conditions that you see on the top for Colossal macropomum, one of the Paku that I studied, to this extreme underbite conditions such as Cataprine mento, which is the lepidophagous or scale-eating wimple piranha down here at the bottom. They are the bane of any orthodontist. I wrote the joke in there. I'm going to say it. It's got to get said. This past summer, I was fortunate enough to have a rock star in REU who found that, like morphology, these degrees of occlusal offsets are likely indicative of the diet in the same way that we understand tooth morphologies have to be closely related to dietary function. But the question of how these teeth and these kind of offset jaw positions are used to break down the mechanically challenging foods that make up the diets of these fish still remain. So I continue my digging, and here's what we know. Of course, we know that all chews are not created equal. You're going to have mammalian carnivores that aren't going to do extensive processing. The tuatara, which is the non-mammalian animal that is going to extensively process food to the point that it's been called chewing. Stingrays, pigs, and giraffes that all have very interesting lateral translations of the jaw to break down their food as best as possible. And while mammals are seen as the definitive chewers as a result of their translational ability, there are still going to be a few other niche traits that make the relatively complex feeding system of mammalians its own thing that has been termed mastication. But we're still finding complex prey crossing behavior in these paku fishes. And so what you're seeing here in this video is the red belly paku consuming a shelled almond at its oral jaws with what can definitely be considered some kind of a relatively simplified feeding system when compared to the complexity of mammals. These fish also have rudimentary pharyngeal jaws, and so outside of what your typical teleostean or spine-finned fish prey's processing condition, they extensively break down their foods at their oral jaws rather than at the jaws that are in their throat with these incisiform teeth of nightmares. Now, because they're limited in their traw translational ability as a result of a almost quintessential like hinge joint at the quadratomandibular, and because they don't have the number of muscles that are associated with feeding that mammals have, such as the temporalis and the masseters for both a deep and a superficial layer, how are these fish consuming fruits, nuts, and seeds, and even more impressively, leaves and grasses that we are constantly eating when they don't have the same tools as what is undeniably the champ of feeding mammals? When you have such a light bag, how do you overcome food mechanical properties? To get at the question of how these fish accomplish that task, what I use is a technique that was invented here at Brown under my professor and Dr. Steve Gatesy called X-ray reconstruction and movement morphology. And it lets us understand this feeding system at a level of resolution which would be difficult to attain through external methods alone. And while a lot of you probably know this technique by now, I'll still give it a brief introduction as I have like my family and friends here checking out this talk. So by combining biplanar high-speed X-ray videos with marker tracing and CT scanning, we can create 3D animations for visualizing rapid skeletal movement of these different organisms in vivo. Hello. What you're seeing here is the same fish in the earlier video chewing an almond using the X-ROM technique that I just described. And I'm going to run you through the bones really quickly because they'll be a bit more important to this talk as I go on. The blue that you see at the top there is going to be your neurocranium, which I colored in a new denim blue because they have the best Crayola colors on Adobe Illustrator. The mandible is going to be mango tango, that kind of orange looking color right there that's chewing those two beads that are embedded in the almond. And the suspensorium, which is a series of multiple bones that connect the lower jaw to the neurocranium, you'll see in a lovely tropical aqua. Now I'm going to show this video again from the lateral view so that you can see the fixed motions of the, oh, one second. Now am I doing it? Yeah, there it is. In the lateral view, so you can see the fixed motions of the suspensorium and the jaw. And if you watch the video closely, I want you to pay attention to the magnitude of rotation at the lower jaw. What you're going to see is that there's two distinct biting behaviors, which I'll break down a bit more thoroughly now. First, you'll see these quick, small bites where food is merely compressed and what looks to be kind of these random repeating jaw closings, closer to that easy end of the spectrum where it's not necessarily chewing, but it is food breakdown. We've termed those compressive bites. On the other hand, there are also going to be these more exaggerated chews where food reduction actually takes place during the fooding bout. And these large bites are going to apply shear force to the food item and result in the edges of brittle foods being broken off, effectively reducing food size for transport in what can best be termed the shear-like power stroke. I previously called this an overbite shear, but because there is an actual overbite shearing behavior seen in freshwater stingrays, and they're not the same thing, we've since changed it back. So if you went to SICB and you wrote down overbite shear, I'm sorry, it doesn't exist in this species anymore. We're learning. Here I have the two different bite types side by side so you can see them more clearly. On the right, like I said, that overbite shearing behavior, as opposed to these random compressive bites where food is merely just kind of repositioned for these shear-like power strokes. Here we have a trace of the motions of the mandible about an axis through the quadratomandibular jaw joint, or the jaw joint that I said is restricted to just those hinge motions. And without having a definitive way of differentiating the two chew types at this point, I went through my videos and visually determined where food appeared to be compressed or dragged, as opposed to kind of power stroked here. And then I've identified the peaks for you to see. If we isolate and zoom in on a particular region, we can compare the plotted rotations with the corresponding X-ROM animation and see these two bites in the context of a continuous trial. Here we start to notice more of the differences in the positioning of the food, as well as the time necessary to perform each bite. But as further tests of this trial, I want to understand if the utility of the overbite is going to allow for a reduction of that space between the upper and lower teeth. So we check the dorsal ventral as well as the rostrocaudal motions of the lower jaw by placing a landmark at the symphysis of the mandible and tracking the movement of this landmark relative to an anatomical coordinate system set in the neurocranium. And it gives us a trace like this. The plot's going to show the displacement of the landmark that we placed at the symphysis, this yellow dot that you're seeing on the fish here if you're looking at it rostrally right on. And the plot's going to show displacement of that landmark for the full trial, and each different point represents a position at a different timestamp. I assigned 0 for this rotation at the jaw joint, where the lower jaw was in its natural resting condition, which for these lovable idiots is slightly ajar. And when I went through and assigned a color scheme for those bites that were the power stroke as opposed to compressive stroke, we're going to see that those power strokes in red are going to far outmove those compressive chews and closely cluster those different motions of the power stroke together. That way the jaw can more closely break down the food using those shear-like forces that I previously talked about. While presenting my findings at a conference in July 2018, my house was unfortunately robbed here in Providence. Police took 3 days to come to me, and when they did come, basically the only advice they gave me was, we suggest you upgrade your security. Led to problems with my landlord, who eventually tried to evict me, as he felt I was being both difficult and aggressive. This was during the course of my qualifying exam. So I was testing for normal chewing behavior, and we're looking to see if the overbite shears, which I've termed as the power strokes, I apologize for not changing that label there, I see it on the right side now, if it was significantly different from those compressive bites. And we see exactly that. Those power strokes are their own unique thing, and it can further be exemplified through EMG recordings, where a previously collected EMG trial from Dr. Fran Irish further highlights differences between the two bite types, as muscle activity in the power stroke is recognizable by pulse muscle activity in the second division of the adductor mandibulae, as well as the levator operculi. Now, if we change the name of the adductor mandibulae second division to match with the current phylogenetically conscious nomenclature put out by DeSavo a couple of years back to allow for more accurate tracing of the evolution of the adductor muscle complex, we see that the pulse muscle activity of the adductor mandibulae is really going to be the pulse muscle activity of the pars rectalis muscle. Hubie et al. Compared the divisions of the adductor muscles between the Paku and its carnivorous relative, the piranha, and found the pars rectalis of the herbivorous species is significantly larger in what may bear some connection to this pulse muscle activity of those power strokes, further setting the power strokes apart as its own distinct behavior. The only problem is that we're still not entirely sold that the power stroke motion driven by the action is driven by the actions of the adductor mandibulae muscle alone. So that very large cheek muscle might not be the only thing that's contributing to these power stroke behaviors. SEM images reveal, oh, it's a bit blurry, but if you look at the left tooth here that I'm circling with my cursor, I hope you can see that, you'll reveal that patterns of wear at the premaxillary symphysial teeth, or the teeth on the upper jaw, are incompatible with food-induced wear, suggesting that this wear pattern that you're seeing here in the inset was brought about most likely by some kind of self-induced wear associated with tooth-to-tooth contact. About the same time that I was finding this all out was around the same time that Botham Jean was unfortunately killed in his home. The officer was convicted. As I was saying, I had absolutely no idea how those tooth of the lower jaw could possibly inflict wear on the upper tooth row, given the degree of occlusal offset present in these fishes. And while I can go on forever about the degrees of occlusal offset and serous salmons, I think the primary idea that we should talk about is the morphology of the rounded suspensorium in these fishes, or that joint that would permit rotation of the suspensorial joint to offset this difference that we're seeing between the lower teeth here and the upper teeth here. This idea was primarily based on the anatomy of the structure alone, as we see a very curved neurocranial hyomandibular joint, which is going to be different than your stereotypical teleostean joint, where the hyamandibula of the suspensorium region here that I'm highlighting is typically going to be tripartite or bipartite with a very prominent strut to stop motion in that rostral caudal position. In fact, it's so stopping, according to a paper by Winterbotham earlier, it's defined as almost a V strut in that the two of them prevent motion in the rostral caudal orientation. So moving backwards and forwards from the fish's nose to the fish's tail. The two suspensorium being on either side of the head, so in bilateral. This is my work from this year, and as we all know, about the same time as the murder of Ahmaud Arbery. I was talking about the motions of that rounded suspensorium, and here we have the trace of the motion of the hyamandibula rotating about a red axis whose placement I've shown here. These data don't really say much by themselves when we overlay these suspensorial data and normalize the magnitude of those rotations relative to those rotations about the quadratomandibular jaw closing joint. Interruption again to talk about the death of George Floyd, and where a police officer kneeled on his neck for 8 minutes, and I think it was 46 seconds. I apologize for the mistake. But yes, we see that the peak suspensorial rotations are right in line with those identified for those power strokes at the lower jaw. However, while this is still exciting, it might be inaccurate to say that this finished the story all the way and ties up a pretty bow, because we are still unsure of the mechanism behind the suspensorial rotation. The adductor mandibular complex, although it's a single muscle in these fish, is still a very highly divided muscle. So for future directions, I'm definitely going to have to further understand exactly what is going to cause that rostral caudal rotation that we're seeing as the jaw is protruded forward at the same time as those power strokes. But the most likely thing that I can come up with right now is that the adductor arcus palatini, a muscle that's going to insert from that hyamandibula, which is at the neurocranial joint, to gain that rotation that I was talking about, the rounded joint, because it only inserts in the mesopterygoid within this Paku species, it's likely going to be the thing that's causing that rotation in that direction. In other fishes, you're going to have connections of that adductor arcus palatini to the metapterygoid as well as the palatine, and within the piranha, like I said, the carnivorous relative of this species, you're seeing insertions of the arcus palatini into a primordial ligament, which has been hypothesized to permit this rotation. And with that, I have no more to say about my research, not really much getting done during the quarantine times, but I think this is enough time to address the elephant in the room. I've put these things up because this has been a very unique time in just America in general, where for the first time, the experiences of Black males and just Black individuals in general are being acknowledged and recognized largely in part by most of the country. You can talk to any Black grad student and they can tell you some of the same things that they've had to deal with. The Black and the Ivory stories, they hurt to read, I'm not gonna lie to you, but I thought it was good for those stories to finally get out. And up to this point, everything I've been saying has kind of been scripted to some degree. I do things off the cuff where I can basically write down a general thought that I need to hit with every slide. But as I talk to you right now, I'm really just talking straight up. It's hard, man. Being a graduate student, I study fish when people that look like me are getting murdered by the people that are supposed to protect them. And I'm not sure if this is the place that I'm supposed to talk about it, but I go to all these diversity and inclusion trainings, and there's things that I want to say, but I never get the chance to say because it's not really my place to monopolize those meetings. But it's hard not to take up so much space when you are one of two Black people in your entire department. When you go to conferences and nobody looks anything like you, it's rough. You don't really feel welcome. And it's hard to know that you have all the resources and the people available to help you get to where you need to go. But at the end of the day, you still have so much shit that you have to fight through. And I'm sorry for saying the S word, Ma. I love you so much. But it's just how I feel, real talk. And so when I was asked to do the all FHL talk, I knew that like immediately I wasn't just gonna do my same research talk. I didn't really get any more findings, honestly, between SICB and now. So it was kind of a, what am I gonna talk to these people about? They all know me, they all came to my talk. So I used it as an opportunity to really just open up because a lot of people have reached out to me and I haven't answered everybody. I do appreciate the well wish that I've gotten from the FHL community in general, and everyone else who's tuned in. And I really wish I could tell you that I knew what I wanted from everybody, but more than anything right now, I just wanted to get this off my chest. I have done like all of my work while going through stuff that half of y'all probably can't even imagine. And we just kind of keep our head down and keep it moving because that's like what the Black experience is. But it's nice that people are finally acknowledging that the shit is a little different. And with that, I will finish my talk and not hold up any more of your time. Feel free to ask me questions about all these different experiences I put up, as well as the research, or if you just have any kind of general questions about where I'm going now with the rest of this Sarah Salmon research, because it is still very interesting, and please be on the lookout for more from us in the coming months as I am about to publish my first chapter working closely with Dr. Brainerd. Thank you for your time.